I divided the chapter on renal disorders into 6 lectures. Lecture 1 is just a review of the gross anatomy of the renal system, the nephron, and urine formation. Lecture 2 is polycystic kidney disease. Lecture 3 is pyelonephritis. Lecture 4 is acute glomerulonephritis. Lecture 5 is chronic kidney disease. And Lecture 6 is acute kidney injury. I think it's important that you listen to the lectures in this precise sequence. I didn't exactly follow the sequence in the book. I followed what was logical in my mind, which is the complication of the first 3 disorders, which are polycystic kidney disease, pyelonephritis, and acute glomerulonephritis, especially if not managed well, is chronic kidney disease. Thus, the topic chronic kidney disease follows these acute disorders. In chronic kidney disease, which is lecture 5, I also discuss dialysis. The last lecture, lecture 6, is acute kidney injury. I start by differentiating it with chronic kidney disease, and I go into more detail on the 3 different types of acute kidney injury. So let's start with lecture 1. This is lecture 1 of 6 lectures on renal disorders. This is a brief review of the anatomy and physiology of the renal system. By the end of this lecture, you should be able to identify the parts of the renal system, describe the nephron, discuss the process of urine formation. Here you see the renal artery that provides blood to the kidneys. Here. The kidneys are bean-shaped and the size of a clenched fist. I've seen a real kidney when I witnessed an autopsy, and I would say it was a bit smaller than my clenched fist. And then you have the ureters that empty into the bladder, and then you have the urethra, which empty out urine out of the body. Now let's take a closer look at the kidneys. If you were to dissect or cut open the kidneys, you would have 2 major regions. The outer region is called the renal cortex, and an inner region called the renal medulla. And you can see the renal artery. The renal artery delivers blood to the kidneys, and that's important for you to remember. If you were going to do a microscopic view of the kidneys, you would be able to see the nephron. Each kidney has about 1 to 1.2 million nephrons. The nephron is the functioning unit of the kidney. So what is the functioning unit of the kidney? The nephron. Let's look at the nephron with our pretend microscope. Here you can see the anatomy of the nephron. It starts with the afferent arteriole, which carries blood toward the Bowman's capsule, while the efferent arteriole carries blood away from the Bowman's capsule. The Bowman's capsule is a funnel-like structure that envelops a tuft of blood vessels or blood capillaries, which is the glomerulus. Fluid flows through the Bowman's capsule into the proximal convoluted tubules, down into the loop of Henle, through the distal convoluted tubule, and into the collecting duct. So what I just described to you is the path of urine formation. So let's discuss the process of urine formation. As you can see, there are four steps of urine formation. Number one is filtration. Number two is reabsorption. Number three is secretion. And number four is concentration. In this slide, let's focus on step number one, which is filtration. Focus your attention on the two arrows at the top. One arrow is toward the glomerulus and the second arrow is away from the glomerulus. Below these arrows are the afferent arteriole and the efferent arteriole. If you notice, the afferent arteriole has a wider diameter. In fact, a much wider diameter than the efferent arteriole. These differences, or this difference in diameter, creates hydrostatic pressure, which makes filtration possible. The other principles that make this filtration system work are size and polarity. Imagine the lining of the glomerulus and the Bowman capsules have holes or fenestrations, but these holes are tiny. This filtration system will not allow large molecules or cells to pass through it. For example, a red blood cell will not be able to pass through these holes. Thus, urine should not contain red blood cell in a normal non-menstruating adult. Negatively charged particles such as proteins will be repelled and are not likely to pass through it. Small amounts of protein in the urine is normal, but large amount of protein in the urine points to kidney damage or damage to the glomeruli. You must remember that the basement membrane uh, is negatively charged, so it will repel negatively charged proteins. So I just told you about the first step, which is filtration. Filtration is based on hydrostatic pressure, size, and polarity. The second and third steps are reabsorption and secretion. Reabsorption means returning to the body, and secretion means leaving the body. For example, glucose is reabsorbed here, but the body has certain threshold for glucose, 220 milligrams per deciliter, above which glucose cannot be reabsorbed, and you can then find glucose in the urine. So glucose in the urine is not normal. Major substances that are secreted are urea, uric acid, and creatinine. That is why with renal function decline, urea and creatinine levels in the blood also will rise. Major ions that are secreted are hydrogen here and potassium. With hydrogen, you can see why kidneys play a role in balancing pH, because pH is just a measure or a metric of hydrogen concentration. And the last and fourth step in urine formation is concentration. The loop of Henle plays a major part in urine concentration because it is most permeable to water, and thus a lot of water is reabsorbed into the body in the loop of Henle, which leaves the urine more concentrated. So the yellow pertains to reabsorption and the green pertains to secretion. This is the end of lecture 1. Please refer to chapter 61 to review on the anatomy and physiology of the renal system.